Okay, so the final topic, which was a completely new topic, but a really, really interesting new approach to understanding the biology of disease and potentially treating the disease, is using the CRISPR gene editing technique. And we were very pleased to have a special lecture at the IMWG Summit from Ern Witta from UCSF, San Francisco. One of the first labs where they've started to use gene editing to knock out unwanted genes, for example, which is in the title of his talk here. And this is one of the first teams actually looking at doing that in myeloma specifically. And so starting to allow us to think about what can gene editing, precise gene editing, mean for understanding myeloma and potentially for treating myeloma. And Ern was starting from the ground up, educating us about the way that this works, and he had rather a beautiful video to show how this gene editing works based upon a natural biologic mechanism used by bacteria to defend themselves against bacteriophages. And so interesting to see that two really smart women were involved in the early discoveries for CRISPR technology and published the early data on this. I think that just to move the discussion forward, if you're going to edit, obviously this needs to be specific. If you're cutting out genes, you don't want it to be cutting out genes that you need, so off-target is extremely important. For some diseases, it might be helpful to treat some of the cells. For cancer, obviously, you need to be able to treat all the cells or a majority of the cells. So very, very important to have a highly active method. You need specifically to be able to work with the plasma cells. There is an interest in the cancer biology space where if it's difficult to edit all of the cells, it might be helpful just to suppress selected genes. And so this could be a helpful way forward in the short term. And so Ern presented a lot of different ideas in the ways that CRISPR technology might be used. And so this is a new approach. Sagar, how do you view this? Is this early technology, exciting technology? How would you characterize it?
Yeah, I mean, I think there's no question that the CRISPR system has had a tremendous impact on the laboratory side of what we do already. And for the last 4 to 5 years, this has become a major part of many laboratories in trying to understand what are the genes important, for instance, in developing drug resistance. So right. CRISPR screens for bortezomib resistance, CRISPR screens right. for daratumumab resistance. All of those kinds of things I think are really important and have been done by some really key scientific people in the myeloma community.
Right.
The role that Arun really showed us was how can we potentially use this in therapeutic potential targets. And I think it's very early right now to understand exactly how we're going to do that. But the idea of being able to knock in or knock out certain genes, whether it's in a CAR T cell, so a cell that you've manipulated to over yes. or under express certain things, or whether you're trying to go after a tumor cell and yes. sort of normalize its dysregulated function.
Yes, or maybe upregulate CD38, for example.
Correct.
He gave that example.
Correct, yeah. Those are all things that I think we'll have to see more modeling, but it could be a really exciting concept.
Yeah, so Joe, what do you think?
You've summarized it well. I mean, myeloma is not as simple a disease as some of these others where the genetic pattern is a little bit more clear and we can always target that one area and repair it. You know, I think CRISPR will have a greater impact in some of the other diseases where the genetic pattern is a little bit more consistent or predictable or thought through or discovered where
it's just already being used in some of the hereditary
Right.
diseases
Correct.
like muscular dystrophy and
Right.
sickle cell anemia,
Right.
actually,
Yeah,
where
yeah.
you
Or
can
You don't need
correct,
100% correction.
but you don't
Correct.
need 100%
Right, and
correction.
even some of that correction can really have an impact. And some of this is really, I think, it affect the pediatric population. We're going to see there's even some, early work in utero trying to look at, affecting a child before they're born who
Absolutely.
may now not be able to have to be born with hemophilia or sickle, at least to the degree that they would have otherwise. So I'm not saying that it's not exciting. I think it's very exciting technology. And again, we want everything available to us. A major role for CRISPR and myeloma, maybe down the line and a long way off. I think in the short term, almost like we thought of maybe CAR T-cell therapy 5 years ago, and it was kind of just coming out. Let's wait, let's see how it evolves,
Right.
and we may be able to apply it. But I was excited to have this as part of the summit. I think there was a lot of buzz about it amongst the individuals here for the summit because we want to know what's cutting edge. No pun intended, cutting. But we want to know
cutting.
we want to know what's coming and how this could influence our field.
Right. And I think, as you emphasized at the beginning, in the lab, I think it's really having a huge impact on our ability to understand resistance, driving mutations, for example, and we're excited to use this as a tool in the iStop-MM program in Iceland, where we're hoping to identify some key earlier driving mutations. But you identify mutations, but what do they mean? And so looking at that back in the lab, knocking the things in and out, you can start to understand the meaning of some of these mutations, probably multiple mutations that will need to be analyzed.
Yeah. No, I think it's exciting because it's a glimpse into the future, and I think that's always reassuring for us to see.
Absolutely. So, well, I think we're getting towards the end of our allotted time here. So, any last words? I mean, from ASCO, from the summit, moving into EHA, what is the hottest area that you think has emerged that is being discussed? What do you think is the...
I think probably looking at the abstracts from ASCO and again this week at EHA, I still think CAR T-cell therapy is probably now the greatest interest. To see from the BB2121 study, as we had now like over 40 patients presented in that study, as we start to see more of them come out, will there be a bit of a consistency around the progression-free survival and how people are tolerating it? So I'd probably say that's still the hot buzz, but there's still, as we've mentioned, a lot of other things, including the newer drugs, the selinexors, the venetoclaxes, the cetuximabs, that'll probably be the next ones in line for approval.
Right, I think without doubt. Well, at our summit here, when we said who's going to go and discuss in which group, everyone was rushing for the CAR T door. I think that it was pretty clear that people were excited to learn more, but also, as you were mentioning, particularly Sagar, to understand better some of these things. What kind of cells are those? How many do you need? What are all of the variable factors related to this therapy, which we are just starting to scratch the surface, really.
Yeah. I mean, I think, again, it's the half full, half empty kind of discussion. The CAR T cells are a great option for patients who don't have a lot of other treatment options. But I think it's important to also realize that there are a lot of patients that are doing remarkably well for a long period of time.
Absolutely.
And that's good news for everybody.
Yes, and we're seeing more of that, long survival with these different combinations, which we talked about, especially DARA combinations.